Hi everyone, my name is Alexis and I'm an undergraduate researcher from Wittenberg University working with the Duke University Marine Lab. My project is titled An Analytical Assessment of the Spatial Dynamics and Population Structure of Atlantic cow-nosed rays during their northward migration. Cow-nosed rays are migratory elasmobranchs that travel in large groups called fevers along the United States Atlantic coast each year. They play an important role in coastal ecosystems, but despite being so visually striking, many aspects of their migratory behavior, like how they move, organize, or separate by sex, are still poorly understood. Most past studies have tracked migration at broad scales, showing that cow-nosed rays move north to the Chesapeake Bay in the spring for breeding and south again to Florida in the winter. But we still know very little about their local scale behavior, such as how far apart fevers travel, which direction they orient, or whether males and females occupy different habitats during migration. So the purpose of my study was to look more closely at the fine-scale spatial behavior of migrating cow-nosed rays, specifically how their group structure and movement patterns vary within a small region of the North Carolina coast. I used aerial imagery collected by drone over Core Sound and South Core Banks, North Carolina, during April 2017, when rays were actively migrating north. My objectives of this project were to quantify fever size, direction, and spacing between groups, examine whether males and females were distributed differently, and investigate how group size and location might influence movement trends. By understanding these small-scale patterns, we can get a clearer picture of how cow-nosed rays navigate complex coastal environments and how changing conditions might affect their migration in the future. To analyze their behavior, I selected 41 drone images from 10 different sequences that showed clear groups of rays in motion. These images were processed in morphometrics and ImageJ software that allows for accurate spatial measurements. For each fever, I measured its overall area, length, and width, the average disk width of individual rays to estimate sex composition, the distance traveled between sequential images, the spacing between neighboring fevers, and the direction of movement after aligning each image to true north. By comparing these variables, I could determine how fevers were arranged and whether environmental location, such as offshore versus nearshore, correlated with group size or sex composition. Moving on to the main findings, first, when I looked at disk width measurements, I found clear size differences among image groups. Some sequences had smaller average widths, around 2.3 feet, which aligns with the expected male sizes. Others had larger averages between 2.6 and 3 feet, matching with the expected female sizes. This pattern showed that most fevers were female-dominated, especially those located farther offshore near Horse Island, while male-dominated fevers were found closer to shore or in marsh areas. This sex-based segregation likely reflects different behavioral roles, with females seeking stable, resource-rich waters for gestation and males remaining near shore to feed or search for mates. Next, in terms of movement trends, I found that most fevers were either expanding or pulsating. In other words, they were actively changing shape as they moved. Larger fevers tended to expand, suggesting dynamic, coordinated movement in open water, while smaller fevers pulsated more often, perhaps responding to changing local conditions. When looking at directionality, half of all fevers were oriented northward, consistent with the timing of their spring migration. Several others faced northeast or northwest, indicating subtle shifts between offshore and nearshore zones, likely tied to reproductive or environmental cues. Finally, spacing between fevers varied widely, from as little as 6 meters to as much as 75 meters. Tightly grouped fevers were usually offshore and female-dominated, while widely spaced pairs tended to include males in more variable habitats like marshes. Altogether, these results highlight how environmental context, sex composition, and group size interact to shape how cow-nosed rays travel. These findings align with previous research showing that female cow-nosed rays dominate offshore during spring migrations toward the Chesapeake Bay. The offshore movement likely helps them avoid predators, minimize male harassment, and assess stable conditions favorable for gestation. Meanwhile, the presence of males near shore and in marsh systems suggests a more opportunist strategy, which is feeding, waiting for females to arrive, or conserving energy in calmer waters. The variation in spacing and movement also tells us something important. Migration isn't uniform. Even within the same region and time frame, different groups can be at different stages of migration, some resting, others traveling, and some reorganizing into new formations. By understanding these small-scale behaviors, we can start identifying critical stopover areas that support the species during migration. Protecting these regions, especially those near Core Sound and the southern Pimlico Sound, could be vital for conservation, particularly as climate change and coastal development alter key habitats. In future work, I'd like to pair drone imagery with environmental data such as water temperature, depth, and salinity to see how these factors influence group positioning. Incorporating GPS or acoustic tagging could also help